It's a pleasure to be here, and I definitely feel that this center keeps on growing, and it's definitely reached a critical mass now of people and resources that I don't think is rivaled by very many places at all in the world. So it's really a pleasure to be here. So what I'm going to talk about briefly is a little bit about resting state fMRI. And my group at the National Institutes of Health, I have a section on functional imaging methods that investigates details and mechanisms behind fMRI, and also develops methods for extracting information. And one of our focuses right now is on resting state. And I also run a core facility, which a lot of people actually use resting state. And just to briefly, what I'm going to talk about is a brief introduction to resting state. Secondly, what does the time series actually contain? A little bit about our approaches towards noise removal, which is a big problem in looking at resting state fMRI. And then how we can extract more information. And then lastly, I'll just end with a slide on still remaining open issues. Even though resting state has really taken off in the last 5 or so years, I believe it's a little bit dangerous, because I think that you get your signals too easily, and it's almost potential. There's a potential for over-interpreting the data as well. So I just want to start with this. This was a recent, really nice ceremony in Milan just last week, and it was with Magnetic Resonance Medicine. It's a journal that's 30 years old. And it was looking at the 30 papers in magnetic resonance medicine that shaped the field. And one of the papers was Bharat Biswal's 1995. Bharat is actually the person who is right here. And this paper actually was published in 1995. And it was interesting when it came out. I remember when it came out, actually. Bharat was, he had this processing platform called AFNI. It was a very early version of it, in which you could actually pick a voxel to use as a reference function. And we just use it for function. And he decided to actually use that just looking at resting data. And that was the innovation, just simply having this platform that lent itself to some exploring of the data that allowed him to discover this. And he did this, and he found that during rest, areas that were correlated also appeared functionally organized. And it was interesting, when that came out, everyone kind of ignored it for about 5 years or so, really, truly ignored it. And then people thought it was an artifact. They were saying, oh, it's probably a scanner artifact or it's related in some way. And then suddenly, in about 2005 or 6, it exploded. So here's just briefly, just for those in the room who don't know what resting state is, just a very quick introduction. This is a typical task activation fMRI. You have left finger movement in blue, right finger movement in red. This is switched. And you can actually see that they're anti-correlated, and they show strong activation with the task. Here, the subject is lying in a scanner doing absolutely nothing. The data look like noise, but if you look at the correlation between a voxel and, let's say, the right motor cortex, the left motor cortex, it correlates with areas that seem to show a similarity to the motor cortices. And so that was one of the first findings. And then since then, we've found these sort of correlations all over the brain. And so the question is, what does it mean, and how to actually do the processing of the data? So there's many different ways of looking at this data. And that's actually one of the interesting aspects of fMRI, is that we actually are still trying to figure out exactly how to get our heads around this data. An initial study by Mike Gracious actually demonstrated an example of the default mode network. It's a well-known network. If you put your seed voxel in the posterior cingulate, you get this typical network that's active, but it's more active during resting state than during a task. And another much more efficient way of actually looking at the data, a little bit more of a black box though, and so it's open for interpretation, is ICA. And this is some work by Steve Smith's group at Oxford, actually doing some preliminary ICA results where all the networks sort of naturally fall out, looking at the spatial independence. And you can do other things too. And this is some of our own data looking at making movies of our seed voxels. So basically, if we move our seed voxel, this is in red right here, we can actually see along the left motor cortex, we can see that corresponding to the right motor cortex, both with two different ways of displaying the surface data. And what's interesting, actually, is that it seems that right around the primary hand area, the correlation goes down, which seems to correspond to anatomic data looking at the lack of connectivity between the primary hands areas. So there's some relationship to connectivity as well that we seem to be gleaning from this connectivity, this resting state correlation data. And another way to look at it that's even more efficient potentially and more powerful is to chop up the brain into segments. Instead of having 10,000 segments of voxels, we chop up the brain into, let's say, 100 segments, 100 regions, and we just make a correlation matrix. We put all these regions in a time series and make a correlation matrix of the pairwise correlation between each region and every other region. And so this makes for a nice pattern that we can actually do analysis on. That's basically a time course correlation matrix. So why is resting state important? First, number of papers has exploded. And this is an example of that. This is looking at, over the years, looking at number of papers in fMRI and a number of papers in resting state, and it doesn't really completely do it justice, but it starts to completely take off around 2006. And this is actually looking at the fraction of papers that are using resting state relative to the total fMRI papers. And this is just completely on a complete upward curve that's right now approaching 20%. I'm sure it will potentially exceed 50% in a couple of years. So that's pretty exciting and a little bit scary too. People are finding things with resting state, and it seems like it's almost too easy to find things in resting state. There's neuronal, psychiatric, and developmental disorders that seem to show differences in those populations than with resting state, than with normal populations. So they're pulling out all these interesting differences that occur between populations, also occur between cognitive states, and so there's a lot of potential information to be had. The methods themselves are in their infancy and they're rapidly evolving. We're still trying to get our head around it. And the neural correlates of these spontaneous fluctuations are not fully understood. There's many studies actually trying to look at the correlation between single unit recording, multi-unit, MEG, EEG, and so on with resting state, but we still don't completely understand why such slow fluctuations. So these are on the order of 0.1 hertz. They're even lower than what's limited by the hemodynamic response. So the hemodynamic response limits you to 0.2 hertz on down. These are 0.1 hertz and less. And so it's not necessarily just because we're looking at a slow hemodynamic response. There's truly slow fluctuations. So what does the time series contain? So this is the biggest problem in looking at resting state. It contains a lot of things. So there's blood, brain, and CSF pulsation. There's vasomotion. There's a breathing cycle. With every breath, B naught shifts in the brain. Subject movement, bulk movement, so on, drifts, scanner instabilities, changes in blood CO2 with changes in breathing. And this is a significant problem as well, that as you change your breathing rate, you have a change in blood CO2, which actually changes the overall signal as well, the global signal. There's changes in vigilance. People have found that the resting state times the global resting state signal is very much a function of how vigilant you are. So if you're falling asleep in the scanner, suddenly your resting state will oscillate, and that will look like something's happening. And also, of course, what we care about, we want to get rid of all of the top part and only look at, we would love to just end up with a time series of spontaneous neural activity. And I have some preliminary data that that were presented for ISMRM that shows that potential. So this is another way of putting it. This is some nice work by Jeff Dunn's group showing how much other things we have to look at. So this is, for instance, looking at, in the visual cortex, for instance, of everything that we want to get rid of is not in white here. And so this is everything that exists either at a voxel-wise level where there's more signal to noise or a region, where there's less signal to noise, and a voxel-wise level where there's a little bit more signal to noise, but there's still a lot left that we have to look through and get rid of. And we still haven't gotten rid of it perfectly yet. So I'm going to show you just briefly our technique for doing noise removal. And doesn't involve a measurement of breathing or anything in the scanner. It involves the basic idea that bold signal shows a echo time dependence. So the percent signal change, if it's a true bold blood oxidation level dependent signal change. The percent signal change will go up with echo time. And we call this multi-echo ICA. And I'm just going to show you some examples of this. And this is a paper by a really good graduate student of mine, Prantik Kundu, who started this effort. And like I said, the basic idea is that the fractional bold change increases with echo time. So we have to collect several echo times to sort of pull that information out. We can't just use one echo time like we typically do when we collect our data. And then we fit this to a model of what we expect. So during rest and activation, this is a typical relaxation curve. With non-bold, with movement, you might have intercept changes in this signal or other things. With bold, everything is the same except the time constant of this relaxation curve. And so that looks like either, in this case, an intercept change looks like a flat percent signal change. With bold, it's a truly a linear response. And so what we do is we collect 3 echoes. These are our 3 echoes right here over time, and look at the echo time dependence of the signal. And so we break down, first of all, our 3 echoes. We concatenate them and make them into, we pull out the ICA components. Then we find which ICA component shows this growth of the signal with echo time. In this case, this is a region of interest from the default mode network, and you can see that shows a nice correspondence to the echo time. And so that's real. Other areas, so this is actually this is the default mode network right here. It shows this nice correspondence to echo time. And if we just were to sort our ICA components based on how well they fit this model, it actually separates pretty nicely into this curve with this elbow. And you have all this noise or non-fitting components and all these fitting components right here. And the non-fitting components, you can actually, some of them are easy to identify just by eye. So you can just see the edge effects of movement here that's a bad ICA component that doesn't fit this echo time dependence. So we do that. It automatically separates into these nice components that all show this fit to this curve. And it gets rid of these bad ones. So this is pulsation, sinus flow, motion, and things like that. And so you suddenly have the good components and the bad components. And here's what we can do with them. So we can do 2 things with them. One, we can just take our ICA components and start to do analysis on those. And so I'll just show you an example of that. Here's where we take our 15 good ICA components, and then we stack these up just like a time series. So then we can do analysis of, on a voxel-wise basis, the belongingness of each voxel to the independent component that is a component that fits the bold response. And if we do that, it actually comes out quite clean that we can actually segment the brain based on the entire brain, based on on how well each voxel fits with another voxel, or how well each voxel resembles other voxels, not in their time series, but in their belongingness to the ICA components. So we have just 15 images in a stack. So we can, even up to 350 segments, you can actually see a good degree of test-retest repeatability. And so here we segment the entire brain, and using hierarchical clustering as well to sort of get the color coding right as well, in each one of these is also a subsegment showing the interconnectedness of the subregion of the brain. And we didn't use any spatial priors to segment this. This is all purely based on the connectivity, on that voxel-wise belongingness to the ICA components. And there's a lot of detail. I'm just kind of showing this because there's a little bit more detail in this than the map reveals. And if we were to color code it, not based on this hierarchical set of colors, but just simply an independent color for each subcomponent, you can also see that there's a lot of test-retest repeatability. So that's one way of assessing that this is real in that sense, that it repeats itself pretty consistently over time. And then we can start to look more detail. We can actually see that there's some functional correspondence, obviously, to these clusters. There's visual cortex, there's frontal eye fields, there's language areas, and it begins to look like a high-resolution version of a Brodmann map. So we actually are in the process of doing more detailed comparisons between these clusters that just from the entire brain that come out within a 10-minute run. This is actually averaged over 7 subjects, but within 10 minutes of averaging for each subject, and other means of segmenting the brain. It seems like it's becoming quite close. And our goal, though, is to do this on individual subjects, to actually do this on one subject and use as a biomarker for either disease state or for classification of psychiatric disorders or whatever. So instead of having a subject doing a task in the scanner, which is very difficult clinically if you don't have motivated subjects, you have them lying in the scanner, collect this high signal to noise. In this case, with a single subject, it's a little bit more noisy, but it nevertheless might be representative in that sense. And actually, there might be some differences in these segments that you might be able to classify, and that's our goal, to use it clinically. And there's still a lot of test-retest reproducibility here as well, even with an individual subject. And also, there's good correspondence with task. And so this is an example of 3 subjects in which we have them actually perform a task on the top row here, and actually looking at the resting state cluster on the bottom row. And so you can see that there's very close correspondence between toe, hand, tongue, language organization, and also the face area right here. So you find the clusters that are sort of correspond, and not only in location, but in tortuosity and other things. So in this particular subject, they were very left language dominant, left side hemisphere dominant, and also it showed up in the resting state as well. So you get, it's very sensitive to the task indicators. And we can actually, even with, so if we do our, instead of doing clustering on our ICA components, we just do simple, seed voxel analysis, but not, once again, not on the time series, but the stack of ICA components. We get really nice detail in terms of even looking at the base of the brain, which has always been a problem, actually looking at connectivity between the thalamus and Heschel's gyrus, for instance, and also looking at other sort of thing if we do a seed voxel approach. Okay. There's one other thing we can do with our time series, with our clusters. So we can actually, instead of just selecting the good ICA components, we can use these bad components as regressors to regress out the signal, to clean up the time series, if we want to stay with the time series to look at. And this actually, to me, was pretty recent and pretty eye-opening. So this is an example of a raw time series that we all know and love when we look at resting state data. It looks, there's some hints of something going on, but it's, for the most part, it's just oscillating around, and there's some pulsation and whatnot. It's hard to interpret. Okay, this is actually our bad components. These are the components that we regress out that look even worse than what we saw before. So there's some bad components that you can see. This looks like nothing, really. So with what's left over, this was what kind of struck me. This is a raw time series. This isn't a windowed running correlation. This is simply looking at raw single image time series now after we've regressed out all the bad components. And you start to see real spontaneous activity. You see the default mode popping out there. You see the temporal areas. You can actually get a sense of it waxing and waning. And this is, once again, this is not looking at a windowed correlation analysis. This is just raw now time series that's been sufficiently denoised so you can start to see the neural activity. So that was pretty exciting to me. There's other things you can do with this too. Now that you've regressed out all the bad noise, you got rid of all the drift as well. And so you can actually start to look at really slow processes. Like, this is actually looking at typical processes in the brain. You can look at, hormonal changes or very slow responses, like with insulin, with eating and things like that. And so here's just one example as a proof of concept that if we regress out all the baseline bad noise, you get rid of a lot of the scanner drift. This is an example of denoised bold and also what's called optimally combined multi-echo, but not denoised. And you can see this, it's problematic to look at any tasks that are longer than a minute. In this case, it's 150 seconds. It stays a rock-solid baseline. And actually even comparing more subtle differences, like slope differences with a task that, in this case, it's a visual stimulation that was changing in contrast very gradually over time. And you can actually get that pretty accurately over the course of 300 seconds. And with typical non-denoised data, it's hard to remove these drifts without taking out the signal as well. So, and then lastly, you can look at different timings of these slow tasks as well, which also has a rock-solid baseline. So it's actually pretty exciting to do this analysis that's based purely on how much the signal resembles bold contrast, as opposed to using any other external measures or templates or whatnot. So we're keeping on working on that. So now the other aspect that I want to talk about in my remaining time is pulling more information out of this signal. And one thing that we have learned is that this isn't stationary. It's not just a single relationship that occurs, that it stays stationary over time. There's a lot of waxing and waning of the networks and how they interact. And so I'm going to just talk briefly about changes in connectivity over time, how we've extracted the most and least stable connections, and also determining cognitive states from resting state. So here's some early work by a postdoc of mine, Dan Handwerker. And everyone has shown this before. Katie Chang, other groups have shown that if you actually just take two regions of interest, one happens to be from the posterior cingulate in the default mode network, another happens to be just from the premotor cortex, and you look at the time series of those two signals, you see that if you do a sliding window of like a minute, you see that the correlation, it's typically 0 if you average them all over time, but it goes up here, and it goes way down, it goes back up again, it goes back down. Is this random? We've seen it to be somewhat periodic. Here's a movie of this. It looks like, things wax and wane, and this is actually not like the movie I showed before. This is actually a sliding window correlation before we had that other technique. But you see things waxing and waning. And so what we thought we'd do to get our heads around this a little bit better is to, once again, look at this correlation matrix, but instead of looking at all of time, we actually look at segments of time. And this is over the course of an hour of scanning. And if we were to compare, for instance, this is the similarity of these correlation matrices between for instance, the first 30 minutes and the last 30 minutes. And a high number here means they're highly correlated, and therefore very similar. So if you average a lot, they become more similar. And it turns out that there's this point at which they start to become less similar, and not in just simply adding noise because you're averaging less, but in a way that seems to show like a structural difference. So in this case, if you average the first 2 minutes and the last 2 minutes of the time series, you see true changes in this correlation matrix. And so there's this, in some sense, this could be like a time constant, or looking at the whole brain of how much or how rapidly you change your state in some sense. So on the order of 2 minutes, maybe it starts to change on the order of 8 minutes, and so on. If we tried to actually then parse this down and look at the pairwise connections on an individual basis and actually find which ones change the most rapidly and which ones change the least rapidly, we've been able to find this. So this is basically a map of the pairwise connections. And we found that the most stable connections seem to be across each hemisphere, and the least stable connections seem to be localized to either within hemisphere or go through the subcortical areas. So the ones that are changing the most seem to show this sort of pattern. The ones that are the most stable and also significant still are the cross-hemisphere ones, and at least we have this data, but we don't know exactly what to make of it, but at least it's something to start with. So in the last part of my talk, I just wanted to talk a little bit about further steps we're taking to pull out more information from the time series. And so we decided to, once again, look at the time series, and in this case, segment them up a little bit more intelligently. Instead of taking what I showed before, like 100 random segments, we're taking segments that correspond to the specific best-known networks in the brain. So in this case, these are the organized 150 regions of interest organized by network. And this happens to be 5 networks in the brain, from default to motor to visual and so on. And it seems like very clear, and this is where it becomes much more dramatic. And instead of looking at where you can't really by eye pull out how they differ, if you look at over time, you can see 0 to 9 minutes, these matrices look incredibly different. They evolve a lot over time. And so how do we make sense of this? So what we decided to do is impose some tasks. In this case, we have 4 tasks that we put in. One is just pure rest. One is a working memory task. One is staring at a video and just sort of paying attention to when, like a video of like a fish tank or something, and when a fish goes by, they press a button or just, well, actually, I don't think they press a button. They just pay attention to it. And then third is just doing math problems. So they're pretty crude sort of tasks, but at least they keep them in a certain state. And so what we do is we make connectivity snapshots, which is essentially a vector form of this matrix. So this matrix, half of this matrix strung out in a vector form, is this connectivity snapshot. And so then what we do is we can divide this into smaller and smaller segments to get a sense of, okay, if we know what they're doing, does our algorithm that tries to look at the correlation between these connectivity snapshots, does it pull out what they're doing in the time series on an individual basis? Can we just tell what they're doing? And it turns out we can. So we have a clustering algorithm that uses either k-means or hierarchical clustering on these connectivity snapshots. And basically, we have them do the tasks, and it actually reshuffles, and it finds the ones that correspond the best. And it turns out that it works really well. Here's the clustering accuracy, or the clustering quality, as a function of window length. So if we do the entire 180 seconds windowing, we get perfect accuracy. And as we decrease our window length down to 15 seconds, we have still a pretty good accuracy of 65%. And what's interesting also is that it seems to correspond to either, this actually happens to be the 15-second windows. This corresponds, this is actually, the clustering actually as they cluster across the task accuracy. This is when they're doing the accuracy of them performing the task right here versus the clustering accuracy here. And also this is interesting, with reaction time. So the longer it takes them to respond and actually do the task, in this case, they have to, either for the math or the working memory, it seems like the clustering accuracy decreases. So you actually find that there seems to be some correlation with accuracy, not with the algorithm. The algorithm seems like it's working perfectly. It's actually that the person has gone off task or is slightly having more difficult time doing the task, which reduces the accuracy. So in this way it seems like it's, once you get a template of what they're doing, you can actually discern pretty well what they're doing in future in other aspects of the time series. So we're trying to carry this forward, obviously, with more subtle tasks, and also we're actually trying to just determine maybe what task is actually, we're trying to build up elements of tasks to see what their cognitive state is spontaneously, if we can actually do that. Something similar to what maybe Jack Lant's doing, what he'll be talking about with his task activation studies. So to conclude, I just want to conclude with some open issues, though. So it's important to make a distinction between time series correlation and connectivity. That it really doesn't imply connectivity at all. It suggests connectivity, but it doesn't mean that there's a connection there. You could have time series that seem to correlate. It doesn't mean they're talking to each other. It just means they happen to be correlating at that time. Or you could have a reduction in connectivity, which could be due to just a reduction in the amplitude of both signals in the presence of noise, and that looks like a decrease in connectivity, but they may not be any less connected. So it's important to keep an open mind about that. We still don't know exactly the underlying neural mechanism or purpose evolutionarily of this slow correlations. And also, do they vary in more than just how they correlate? In other words, it's nice that we're getting all these correlation maps, but it would be really nice to actually have a correlation or a time series signature from each region. So, maybe the motor region has certain frequencies that give it a signature. It would be nicer to actually get that information as to simply saying, oh, the motor region is there because it correlates together. That's great, but it would be nice to dig more deeply into what aspects of that correlate and why, to separate the regions. And also, it's still an open issue, and I think we're getting closer and closer to this, is actually having time series that have nothing but the spontaneous neural activity in them. And then it gets really fun. Then we start to really start to look at this data. So I just want to leave you with this movie that was, to me, kind of compelling to just simply stare at as it sort of waxed and waned. So with that, thank you.